The overall goal of this procedure is to sort human embryonic stem cell subsets by their cell surface antigen binding of the TG30 and GCTM-2 monoclonal antibodies. This is accomplished by first dissociating human embryonic stem cell or hESC cultures into single-cell suspensions. In the second step, the hESC are stained with the monoclonal antibodies TG30 and GCTM-2. Finally, the cells are separated by fluorescence-activated cell sorting and returned to culture. Ultimately, the colony-forming ability of the sorted cells can be assessed by flow cytometry and phase contrast microscopy.
The main advantage of this technique over existing methods like visual observation is that it provides an objective measure of pluripotency on a cell-by-cell basis for a large number of cells, and this facilitates the separation of the embryonic stem cells from the differentiated cells.
This method can help answer key questions in the pluripotency field, such as how changes in the culture conditions quantitatively affect different human embryonic stem cell and human-induced pluripotent stem cell lines. Demonstrating the procedure today will be research scientists from my laboratory, Zhou, Han Qi and Wang.
To make a hESC single-cell suspension, begin by aspirating the hESC culture media from 2 T75 flasks containing a cultured hESC cell line. Wash each flask with 10 milliliters of DPBS and then incubate each culture with 3 milliliters of EEE.